in terms of cryopreservation, what it then means that is a process of preserving tissues or other biological samples at extreme low temperature. Cryopreservation is a process of storing tissue or preserving tissues or other biological sample at extremely low temperature. So we are preserving tissue and other biological sample at a very low temperature. Does that make sense? And the typical temperature range may be like minus 130 degrees centigrade, okay? So even if you don't remember anything, cryopreservation preserve tissues and cells, okay, at extreme what?
Low temperature.
Low temperature, yes. So what then happens is that, so why are we doing that? Of course, if we're saying preservation, that means we're trying to maintain the tissue integrity. We're trying to make sure that the biological activity of that tissue, when we say biological activities, you know that the tissue can, or cells can deteriorate and all of that. Are you getting me? What it means is that if you take, okay, let's say, if you take a sample, I'm just trying to use something that can help you to remember. If you take a sample on a plain bottle, blood on a plain bottle, and you take a blood on EDTA, okay? Now, provided that the EDTA sample is properly mixed, if you come back after 30 minutes from the same patient, the sample will no longer remain the same, as in when you compare two of them. So the one of the EDTA can be better preserved in terms of if you still want it to behave like a whole blood. Do you understand me? But the one on the plain bottle would have clotted. Does that make sense?
Yeah.
And you can't get the whole blood again. You can only get plasma or clotted cells. So in order to preserve the blood then, to behave as that whole blood, that's why we now put it in anticoagulant. So I'm only trying to say that in this cryopreservation, we are preserving. The reason for the preservation is because we want to make sure that the biological activities, okay, both the biochemical reaction and cellular metabolism are halted, okay, meaning that it is put to stationary. Do you understand me? It has stopped. Stop doing anything again. Do you understand what I mean? Good. So because once you get a tissue, there are a lot of biological activity going on. Remember in histopathology when we talk about fixatives.
Yeah.
So once you get any biological tissues, there's a lot of chemicals, reactions, there's a lot of biological activities, there's a lot of cellular metabolism, a lot of things is happening inside the cell that you are not aware of. And that is why if you don't preserve that cell, what is then going to happen is that that cell will start deteriorating. So from something that is not smelling, it will start smelling. Do you understand me? Because the biological activities and chemical reaction has taken place in such a way that after the whole reactions, the cells are dying off. Do you understand me? So, but in order to stop that from happening, we can then preserve it. So the whole essence of that cryopreservation is to halt these very biological activities, this biochemical reaction or cellular metabolism. So they are all forms of biological activities is a general word. So something like chemical reaction or cellular metabolism is an example of biological activities. So what this extreme temperature, low temperature will do is to make sure that these biological activities, have been halted. It has put to stationary so that it will stop. So what we want to do that as soon as we collect the sample, all we want to examine is what is on that sample as we have collected it. We don't want any addition. We don't want any minus. Does that make sense?
Yeah.
And that is cryopreservation. Happy?
Yeah.
Good. Now, if that is the case, I want you to still remember your, what's it called now? Histopathology. Remember that, I will say this later anyway, but remember that even though we use fixative like formaldehyde, 10% formaldehyde to preserve tissue, as good as that is, I hope you know that it can also affect the tissue somehow.
Yes.
Good. Okay. Just have that at the back of your mind. So let's look at the importance of this very cryopreservation. What are the importance of this cryopreservation, especially when it comes to pathology laboratory as regard to biomedical scientists and all of that. Now, remember that because of that -30, anything that is stored at that low temperature can be stored for a very long time.
Yeah,
So,
longer storage.
long-term storage, yes, long-term
Mm.
storage. So cryopreservation would then allow that long-term storage of biological material without any compromising their what? Viability. Do you understand?
Yes.
Okay. So, and this is essential in preserving, a number of, samples that are not very common, if you understand what I mean. Like in biochemistry, I know why I keep going around like this because I don't know other interviews you will get. But like, no. Hopefully this will be the, sorry, I take my word, but I hope you understand. That in biochemistry, when they get CSF, they don't handle CSF. That's, what I mean by CSF. So good. So they don't handle it the same way they handle other biochemistry samples, because CSF is not one of the common sample you'll be collecting. It's even dangerous to collect it. Do you understand me? Okay. Do you get what I'm trying to say?
Yes, I do.
So what I'm trying to say that in this cryopreservation, most of the samples that are preserved, at least even for that very long time, are some of those samples that are not common. They are very rare samples. Are you getting me? Okay. So the examples are stem cells. So gametes. That's good. So gametocyte, okay. So now embryos and certain tissue. So all of those kind of samples that are not that common. So we can use cryopreservation to preserve them for a very long time. The idea is that long preservation can then help anytime we need it, we can go and get it, because it's not very common. Are you getting me? So you cannot allow it to waste like that. Are you following?
Yes.
Remember, we are trying to define. If you go to interview, they say, what is cryopreservation? That's what we're doing. Okay. Good. Now you've mentioned sample integrity. Obviously, that temperature would then help to maintain the sample, making it to be stable. Okay, put it in a stable state. That way, it can be able to help you to ensure that the biological integrity, obviously, of the cells and tissue is preserved. Okay, and that would then help for accuracy and reliability when you do your test. Are you getting me? Because if they degraded or deteriorate, you can't be able to, it may affect the overall result. Okay?
Yeah.
Yes. Now, another thing is that, which is not really important for your interview, but it can help to facilitate research and clinical application. Okay?
Yeah.
Add that, because it means that with this preservation, we can actually take some of the sample for the research, for confirmatory, for clinical trials, whatever thing we want to do. Are you getting me? Okay. It's fundamental in research and clinical settings, enabling storage of cell line for research, the preservation of transplantable tissues and organs and all of that. Okay. So it's very important, especially when it comes to experiment that has to do with like infertility or fertility treatment, something like that. So you can always, because we are using this process to start gametocyte. Okay. Happy days. So we've talked about what cryopreservation means. We've now talked about importance of cryopreservation. 3 things we've mentioned: long-term storage, sample integrity, facilitating for research, or help for research, something like that. Okay. Good. Now the next thing is, how can this then be applied? What is the application of cryopreservation? How can we then be able to apply this? Now, the application, I want you to go back to remember the things I mentioned, that what we are storing are not common samples. Good. So now the first thing you are going to see there is stem cell and regenerative medicine. Stem cell and is regenerative medicine. Are you getting me? Heard about it, right?
Yeah.
So, okay, good. So, because in regenerative medicine, stem cell regenerative medicine or regenerative medicine is like using a tissue or like this stem cell, okay, to be able to have to activate, a repair body system that has damaged. Are you get what I'm trying to say? So that's regenerative medicine. So now what happened then is that this cryopreservation is crucial for storing stem cell. We already know that, okay? What can then happen? Later, we can use it for what? Regenerative medicine. That's just what I'm trying to say.
Yeah.
Okay? So like in things like leukemia, lymphoma, and other hematological disorder. Are you taking note of this point?
Yeah.
Good. Now, the next thing is fertility preservation. Obviously, you should also know what we've said, gametocide. Are you getting me? Okay. So
Yeah.
we're trying to repeat ourselves, but just to add a little bit more to what we have said before. Okay. So now you find that this process, whether sperm, spermatozoa, or eggs or embryos, they are the cornerstones of what? This assisted reproductive technology known as ART. Assisted reproductive technology. Okay, so what it does then is that even if you forget that, just know that fertility, it helps for fertility preservation in its application. What then happen is that in that preservation, then it can then enable the individual to preserve fertility for future use. All right, the next one is that storage. What can we call it? If we're to use in terms of hematology and blood transfusion, what can we call it?
Bank.
Yes. So this application is cryobanking. So meaning that, of course, many biobanks store a wide range of biological materials, including blood samples, tissue. Basically, that's just what it means now. So all these things are being stored for therapeutic purposes. So when we think in terms of therapeutic purpose, that was why the very first day we spoke, I made you aware that if somebody needs 5 units of blood, 1 unit of blood, that is therapy. Don't just think the person is receiving blood. It's treatment. Okay? That preservation of genetic materials. Preservation of genetic materials. So this cryopreservation allow a long-term storage of genetic materials. Okay? So endangering or genetic. So meaning that with that, there won't be any modification or anything affecting that. Does that make sense? So in application, I've mentioned stem cells and regenerative medicine, fertility preservation, cryobanking, preservation of genetic materials. So let's look at the techniques of the cryopreservation. So there are 3 main things we are going to mention. Okay. One is controlled rate of freezing. Control to regulate the freezing rate, something like that. Regulate freezing rate or controlled rate freezing. Okay. 2 is vitrification. 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 Okay. The last one is cryoprotectants. Okay. If something is at -30, like we said, what do you think will happen to that sample? Or let me put it this way. Go on.
It will be frozen.
Yeah, so that means there's going to be ice crystals.
Ice crystals, yes.
So this control rate freezing will help to ensure that there's no formation or minimize the rate of formation of the ice crystals. Because high formation of the ice crystals on those tissue samples, are you getting me, will damage the cells. Does that make sense?
Yes, it does.
So that's for that. Now, number 2, vitrification. Now, this one I don't need to answer what it means. Okay. So what this one does, we already know about the minimizing the ice crystals, right? Now, what this vitrification is going to do now, it's just a technique that is used to prevent ice crystal formation by turning the sample. So it's going to change the sample into glass-like state. Glass, and I know like your glass. It will change the sample to glass-like state. It's a tissue sample, but it will change to glass-like state. And once it changes to glass-like state, there can be crystals.
Okay.
Does that make sense now?
It does.
Now that I'm explaining it to you, because it's not something I've seen live. Now that I'm explaining it to you, I think what I can remember is in those days, when you have ice blocks, some of them sometimes can be very transparent, very like a mirror.
Yeah.
So that, to me, that is like glass-like.
Translucent or something.
Yeah, so you wouldn't expect any form of crystals, as the case may be, hopefully. Okay. So this vitrification is a more rapid freezing technique. That means it will freeze the thing quickly and turn it to what? A glass-like state. And that way it can prevent the formation of what? Ice crystals.
I think so.
Is that okay?
Yeah.
You should know this one. Cryoprotectant. You're right. It's a chemical compound, that can be used to protect the cells or tissue during the freezing process. Okay.
Freezing process, yeah.
And that protection will help to remember the emphasis of the protection. Now we already know that the freezing preserves the tissue. There's no doubt.
Yeah.
The problem we now have is that method forms ice. Are you getting me?
Yeah.
And that ice formation damages the cells.
Yeah.
So this technique is trying to see a way it can reduce that ice formation. So the cryoprotectant is not just to preserve the tissue, because freezing is already doing it.
Yeah.
What we are now using here, that the chemical compound now, okay, is then used to protect the cells and tissue during the freezing process. Okay? What it does, how does it protect it? It protects it by reducing ice crystal formation and cellular dehydration. Do you understand? So the example of the compound could be something like glycerol or dimethyl sulfur oxide. Now, having known this, the next question now is then, what are the challenges? Because remember, anything artificial is not absolutely advantageous. You know what I mean?
Yeah.
Good. So all these processes we've mentioned, whether it's by cryopreservation or cryoprotection, all of this is we've mentioned, vitrification and all of that. What do you think can be the negative impact, whichever way you look at it? Let's start with cryoprotectant, which should be the obvious one. What do you think can be a negative impact, disadvantage of cryoprotectant? Because they are chemicals, right?
Yeah.
So it should be optimized and moderately used, because the high concentration will do what? Damage the cells. The same way, high concentration of formaldehyde will damage cells, remember? So the high concentration of the cryoprotectant can cause toxicity to the cells. So we need to make sure the amount we are using. Now, the thawing process. The cells are frozen down.
Yeah.
You now bring it out, trying to defrost it, kind of, okay? So that process must be carefully controlled to prevent cellular damage, okay? Why? Because if you thaw the cells immediately, that can cause shock, and that sudden shock can kill the cells. Do you understand me? So the thawing process should take a gradual time, okay? And should not be done rapidly, as that may affect the cells, okay? Okay. Now, the another one is, of course, following the thawing, we now need to make sure that the post-thaw, the cells still maintain viable, are still viable after they've been thawed, okay?